from Microbe TV. This is Immune, episode number 92, recorded on April 22nd, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, how are you?
So this is the first day I walked outside and I was dressed too warmly.
That's not a bad thing.
I'm usually
the trees
not
are in bloom.
too
I'm
warm.
happy. Spring
I'm
is
usually
sprung.
Fine. But today I didn't even wear my gloves, but I had a scarf on, so I have to ditch the scarf. But I need that splash of color. I don't know what to do. Maybe I need a light scarf, one of those really thin ones, right?
A silk scarf.
So joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. I similarly was impressed with how warm it was when I stepped outside this morning. I was pretty excited. What if you had a pop of color of like brightly colored glasses or something?
I do have. Normally I have brightly colored glasses, right? Red. Spider-Man. They're red and blue.
Nice.
Also joining us from Cleveland, Ohio, Steph Langle.
Hello. Hi, everyone. I love colored glasses. As you could tell from this episode, I have my red ones on today.
Yeah, they're pretty. They match your shirt.
Thank you. Yes. And Vincent, I always recognize when you wear those blue and red ones. I love those. Those are great.
Yeah, these are not for screens, though. That's the problem. These are for screens, and they're green. So I went to a glasses store in Manhattan, and really upscale, right? And the lady said, "Oh, for TV, you have to have green." I told her I did a lot of recording. But so that's why I have these green things now.
Okay.
Did you tell her that you had purple and pink behind you?
Probably, but I really prefer
those are
these.
great.
These are really cool.
Those are nice. Yeah.
I have all kinds of glasses here. I have clear ones.
Those are nice too.
And I have blue. I like these. I have red at home that I use when I'm at home.
That's a lot of glasses.
Yeah, I have a lot.
So we have 3 glasses wearers. Do you wear glasses?
I have the old lady reading glasses.
Oh, listen.
But they're prescription.
We all get there.
It's a little easier to see if I have them on.
I mean, I've only been on team glasses for less than a year, but
team old lady eyes.
Yeah, team old lady
I'm
eyes.
not calling you an old lady, but
that's fine. It was an apt description.
Fine.
And anybody who's listening and laughing, just wait. You reach a certain age and your eyes go bad. But I don't need distance glasses anymore because my eyes adjusted, so I only need them for up close.
This is the second try at this episode.
Yes. I didn't know if we were going to tell the audience our secrets.
Why not? They should know how things go.
It works.
We recorded this a month ago, but apparently it didn't record, so we're trying again, and I'm so nervous. It's recording. It says uploading. Which is what it did last time, and when I push stop,
it disappeared.
Everything disappeared, and they blamed me at the company. They said, "Oh, you didn't push the."
We all saw it.
I
totally all saw it.
My colleagues
you're finished uploading.
My colleagues all vouch for me.
Absolutely.
Anyway,
so we just really liked talking about the 2 papers we're going to tell you about today.
2 papers were amazing. You just need to talk about them again.
The 2 papers are pretty cool, so I thought we should just do them again.
They are. So, but if our jokes seem rehearsed, that's why.
Yes, that's a problem. There's only one thing I want to say that I said. Everything else I'll ad lib it. It's no problem. But I did have to go over the paper again. I forgot it.
I did.
Yeah, I kind of did too.
Right. Anyway, folks, if you like these paper discussions, science discussions, we give you real science, the facts. We don't give you misinformation. We certainly don't lie to you.
No.
I even told you that the last time the recording screwed up, many people would say it didn't. But it did. Anyway, we would love your financial support because we do not do ads. I feel they are disruptive to learning. This is a learning experience. So to all of a sudden have an ad pop in for Red Bull, that's what everybody wants, right?
Or pillows or underwear.
Whatever you last bought, they will give you ads for it, right?
Right.
But I just bought that. I don't need to see any more ads. Anyway, you can go to microbe.tv/contribute and you will find different ways that you can help us. All right. So this is a reprise.
Yes.
And with
It's
doing
new
this,
to you.
Yeah, to you, it's new. And we will make it new to us as well.
Yeah.
And so the snippet today is from Steph.
Yes. So the snippet today is by Chuhee Liu and Thad Stappenbeck. They are at Cleveland Clinic Lerner College of Medicine, which is just down the street from where I sit at Case Western Reserve University in Cleveland. And the title in this science article is "A Host-Adapted Auxotrophic Gut Symbiont Induces Mucosal Immunodeficiency." And this came out September of last year, so still pretty new, I would say. Within a year is what we would consider new work. And so the background of this work. So when we think about analyzing microbiomes, we think about next generation sequencing, looking at bacterial ribosomal RNA, or sometimes you'll go even deeper sequencing with metagenomics. And this has definitely, these technologies have advanced our ability to analyze microbiomes, identify host microbe interactions. But there's a challenge with these technologies in that they are not understanding things at the strain level. There's not enough, metagenomics can, but most gut microbes can only be assigned by the family or genus. And so there are just so many unknowns at the species level. And that's because it's very difficult to isolate a lot of these guys in pure culture, to take them straight out of the gut. They may not replicate without their buddies that also exist in the gut. They also may not replicate in traditional culture systems, and you might have to get special culture systems with different metabolic capabilities to be able to culture these guys. So even though most phenotypes are driven by, we can understand that to be driven by polymicrobial communities, ecological interactions, with the sequencing approaches we've had so far, it's been difficult to do that. And so that's really setting the background of what this group is trying to do, ultimately leading to ways that we can manipulate the microbiome to improve human health, which requires mechanism-based studies where you can assign causality. And that's paramount. And we did the data generating phase of the microbiome research. And now we're trying to harness that. How do we do that to understand mechanism so we can develop therapeutics? So an important component of the immune system that we're going to talk about today is secretory IgA. So this antibody is secreted huge amounts in our gut, but all mucosal surfaces and even breast milk contain a lot of IgA. They are produced by local plasma cells that set up shop in the lamina propria of the gut. They secrete a dimeric IgA, which finds its way through the lamina propria to the basal side of epithelial cells. It picks up its receptor called PIGR or PIGR, a polymeric immunoglobulin receptor, which is transported to the surface. And this receptor, which is covalently bound to the dimer, protects IgA from degradation. And this is a point we'll talk about in this paper. IgA degrading microorganisms exist to degrade IgA because IgA can help keep the microbiome in place, in check, from preventing it from going systemic, while also blocking harmful pathogens like viruses.
Yeah. So sorry. We could imagine, IgA being in these mucosal surfaces and being particularly important for potentially blocking pathogen replication, just as Steph mentioned. So it's sort of an important first line of defense. But one thing that's super weird about IgA is there's a decent fraction of people who are IgA deficient. And we don't totally understand the details of how that works and like why some people are IgA deficient and why some people are not IgA deficient.
And we said it was like 1 in 200.
I think that I believe
1
that
in 300.
Yeah.
I remember we had a hard time remembering how many people, so I looked it up, and it's 1 to 300 to 1 in 500.
So I know that I always teach it as the most common immunodeficiency.
Yep.
And we don't totally get how it works, and we can think about reasons why there may or may not have been selection, but there's a lot that we don't know about it. I was sort of thought that was cool when I read this paper both
times. So we
times.
don't know mechanistically why people don't make IgA?
We don't. I didn't realize. I actually didn't totally understand that until the last time we had this discussion.
Yeah. That we don't know the mechanism. Although there's not, from my understanding, it's not a genetic deletion of the.
Yeah. I always thought it was a polymorphism, but maybe it seems like it's not actually a polymorphism, and that we didn't know about it. And I didn't realize that till learning about this paper. But it's sort of cool because they have mice that are IgA sufficient and IgA deficient as well, just like those different people.
Right. And so at the end of this paper, we'll talk about maybe this is the results of this study may be why some individuals are IgA deficient. And there's been a lot of debate. So the existence of IgA deficient people and that they're reproducing and they're surviving puts IgA in this framework of, okay, who cares? Obviously, many people do care, but from the context of what does it do? If it's, if you can live without it and survive, why do we have IgA? Why do we have so much IgA? What's the selective pressure of having individuals in our society who don't have IgA? But I do want to mention, so selective IgA deficiency is defined as having serum IgA at less than a certain threshold, with normal levels of IgG and IgM. But they're not diagnosing that by fecal IgA, which talking to some people who study this, there might be differences in serum and gut IgA levels depending on the deficiency. So this paper is set in the backdrop of that these individuals exist, and they also, as Brianne mentioned, have mice that were either IgA high or low. And what they found, they did this screening where they showed that they found complete degradation of exogenous IgA under anaerobic culture conditions from the feces of IgA low mice, but not from the feces of IgA high mice. However, BSA was their control. This is just like a serum albumin. It was unaffected. So this degradation in the IgA low mice was IgA specific.
Yeah, so it's not that they were IgA low, like genetically, there was something that was degrading the IgA and making them IgA low.
Right. And so they went through a series of investigations to try to identify what bug was doing this in these mice. And you can tell it would be interesting to have them on and learn what this process was. But they found a specific colony that had an IgA degrading activity, but then their subcultures were inconsistent. So they had to do some sequencing, and then finally, eventually came to this strain X, and using antibiotic resistance selection, identified this new genus and species, which they, I think, named. I believe they named.
I think so.
Okay, Thomasiella aminophylla. And we commented last time, Tom. There's two authors with the name Tom on this paper.
Yeah.
So we're like, did they do the initial, isolation? That would.
Yeah. And I remember looking last time, and they did describe the entire genus. Like, this was the first member of the genus.
Cool.
And we also mentioned that if Cindy had done this work, it would have been Cinderella.
Yeah.
Oh, look, she still laughed.
So friends listening, think of if you discovered a new microbe, what would the name be that you would choose for your microbe?
Stephanella, Brianella, Vincent
Langella.
Langella. Frank Langella, runs off the tongue. I like that.
Oh, yeah.
Langella, yeah.
Little tough, but so this one was unique, and so it's a gram-negative, but it had this irregular morphology, and they found that it had genetic adaptations in its ability to make de novo synthesis of N-acetylglucosamine and N-acetylmuramic acid. And that's because the enzymes that make these acids, they're required, and they didn't have them. So depending on the environmental supplementation of these metabolites, determined its ability to have a full cell wall or a regular cell wall. And so this is the definition of oxotrophic, is that you require something from your environment, a metabolite, to produce the building blocks of your cell wall, bacterial cell wall in this case. And they did some other cool things where they looked at the growth under conditions of with or without acetylmuramic acid. And then they found these cool outer membrane vesicles that carried the IgA degrading activity, which was so neat. You think about the way these bacteria talking to each other. And so they wanted to further define how this is impacting the mice. So that was looking at the microbe. Let's look at the host. They co-house IgA low and high mice, and it transferred the phenotype. So this is maybe like Koch's postulates, where you're able to see it in the host organism and then transfer it to another. They saw a reversal of the phenotype when you treated with antibiotics, vancomycin. So it eliminated the T immunophyla, and then restored IgA. So seemingly that connection between that bug causing the degradation. They did some interesting experiments to understand, could this bug do this alone, or does it need helper strains, helper bacteria? We talked about polymicrobial environments earlier. It did, in fact, need helper strains, because when you did this in antibiotic pre-treated germ-free mice, and they did notobiotic mice, which, okay, germ-free is they're not, remind me, what is the difference? Notobiotic is they're derived from their mothers directly into the germ-free, whereas...
Yeah, I thought notobiotic is nothing.
It's like nothing, right? It's the bubble mice.
Yeah. Yes. Germ-free, usually they have them like in the bubble, but they bring them out, but I'm not 100% sure on that.
So varying levels of cleanliness, or lack of microbes. And they couldn't, that bug could not colonize those mice under those very strict conditions, even when you supplemented N-acetylmuramic acid. So this is something that you really could only detect the bug, and therefore the phenotype, in the presence of other bacteria. And correct me if I'm wrong, but they did not really then go find out what those other bugs were. I think that's probably future work.
Yeah. I think that's really hard to do.
Yeah, I think so too. Because I think that it often might be that it's not always the same bug in every situation. It may just be some organism with some particular metabolic capability or something like that. And so being able to sort of look at individuals and see, oh, they all have a methanogen or something, like that was just the first metabolic thing I could think of, but they may all be different members, but they all have the same property in common, can be really hard to determine.
Right. So, okay, it seems like they've pretty convincingly established that this bug is responsible for the low IgA because it's degrading it. Now they want to know, how does that affect the mice's response to respond to pathogens or a chemical disruption? And so they did 3 different models. The first was mice vaccinated and treated. So they were vaccinated for salmonella and then treated with both T immunophyla and an IgA high fecal slurry, and both of those showed reduced protection against salmonella. So you reduce the amount of IgA in vaccinated mice, you're not going to protect as well, showing that it's an IgA-dependent protection against salmonella. They did Candida albicans, and because their rationale was secretory IgA promotes clearance as well, and for this bug, mice colonized with the T immunophyla or the IgA high fecal slurry cleared C. albicans more slowly than those with the slurry alone. And lastly, they did a DSS-induced colitis model. So DSS, it's a chemical-based colonic injury model, and this is similar. They had delayed recovery and greater weight loss if they had the T immunophyla. So if nothing else, I was going to say this definitely shows us that IgA is important and shows us some of the things that IgA is doing. And when Steph asked that question of, who cares? Well, look at some things that happen with this lack of IgA that IgA is doing here. Yes. And there's a lot of recent data. I think it's just understanding the diversity of functions that IgA plays was one. The field had to really wrap its head around that. And then you have to do human cohort studies. And I do think there are more and more coming out now that the individuals, especially pediatric patients, are more susceptible to various pathogens than those who have high IgA.
So I was also wondering whether it would have been good to test some sort of autoimmune disease or allergy that's associated with IgA. Are there mouse models of some of those? I don't know. I'm not thinking of a allergy model that is dependent on IgA. Okay. Most of the work I've observed is that
breast milk IgA is indirectly associated with.
Okay.
But I think it's because it protects against viral infections that then induce asthma. It's kind of like it's an indirect, but it's a good point that would be interesting.
Yeah. The point you make also sort of points out one of the other reasons why thinking about selection of IgA might be hard, because if you are IgA deficient, you might still get IgA sufficient breast milk from your mom. And so at least in the youngest age groups, IgA deficiency might not actually matter in the same way because they're getting IgA from another source.
Right.
So did they check nasal IgA in these mice?
They did not. That would be really interesting. From experience, it's difficult to nasally gavage mice and get sufficient levels, but you could pool the samples. But they did not look at that.
What do you think would happen? Would they be nasally deficient too?
I would think so.
Well,
but
I guess
I don't know though.
if
If it's just this bacteria, we don't know if that bacteria is only in the gut.
Yeah, maybe if it can colonize the nose, but if it's an
anaerobe, probably not. Right.
Right.
But, what kind of mice were these? C57?
Mm-hmm.
I wonder if it's in all inbred mice or just this one. Right?
Well, I mean, it looks like they had these mice that were known to be either IgA sufficient or deficient.
Yeah.
And they didn't really know why.
Do you think it arose in the crossing of these mice somehow?
Well, there are other bacteria that modulate immune responses. I think about SFB.
Yeah.
And that's also transferable. So I don't remember, I didn't look into it, whether they got mice from different vendors or whether this was actually a variability that they noticed within their own colony.
I remember trying to look into that last time, and it seemed like it was variability that they noticed in their own colony.
Which is surprising because at some point you would think it would equilibrate if it's transferred when you co-house.
Yeah. Or I wonder if maybe they had different experiments, different questions that they just had two colonies going on, maybe different rooms and then that room got this bug and the other one didn't, maybe.
I'm now, I guess I hadn't really thought about it, but I'm now going through, they never actually look at serum IgA in these mice. It's all fecal IgA.
I think
so we don't even know if their serum IgA is deficient, if this bug has changed IgA systemically in any compartment, let alone nose.
Yeah, the only time they talk about serum is when they incubate mouse serum with T immunophyla to understand if you see cleavage of other antibodies, IgG, IgM, and I'll talk about it in a little bit, how it's cleaving. So yeah, that would be interesting considering clinically IgA deficiency is a serum-based measurement. Mm-hmm. And so I think it maybe goes back to maybe there are individuals that have sufficient circulating serum IgA, but they have a bug that degrades it in the gut. Therefore, they're more susceptible to gut-specific pathogens.
I don't know how much that the OMVs circulate, which would also be interesting because they did isolate outer membrane vesicles from this bacteria strain and showed that that was also capable of degrading the antibodies. You mentioned that because you said it was associated with these outer membrane vesicles. So if those vesicles get into circulation, they might be able to degrade the antibodies in circulation as well.
Right. So they move on. They look at can T immunophyla induce IgA to itself? And that's really this concept that commensal microbes do induce antibodies specific to themselves. It's not just like polymeric IgA that can bind loosely to a bunch of things. That can bind the specific bug with high affinity. They do see that. Then they move on to ask, can it degrade other immunoglobulins? And it said they did degrade all mouse immunoglobulin isotypes, but to varying degrees. It completely digested IgA, IgE, and IgM, but left IgG with resistant fragments. Mm-hmm. So I guess Cindy brings up a good point. Serum, which I'm sure they have serum, and maybe it's in the supplement and I missed it, but I don't think so. if your hypothesis is true that the outer membrane vesicles go into serum, maybe we'd see a decrease in any of those.
I would think that would not be a good thing.
I would agree.
Because I would think it would make those animals extremely susceptible to a lot of different infections. However, if you're housing them in SPF conditions,
right,
maybe they can get away with that. I don't know.
They also find interestingly that this T immunophilia preferentially cleaves antibodies with kappa light chains, not lambda. So we have the light chains of our antibodies are either kappa or lambda. And they use recombinant antibodies to confirm it's the light chain. So whatever this protease is doing to cleave, it's specifically amino acids in the light chain.
But it also degrades the heavy chain though, right? But it's just, I got confused about this because the first time I read through it, I was like, oh, okay, yeah, just kappa light chain, da, 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 da. But if you look at the gels, the heavy chain goes away. Mm-hmm. So somehow it's when it's associated with kappa light chain, it's more sensitive to this, but it's degrading both the heavy and the light chain.
Or maybe the other way to interpret that is the lambda light chain is resistant in some way. That there's some, and
it protects both.
Mm-hmm. Yeah.
Do you think that I'm working this out of my head, so I don't even know if I totally believe it, but could you imagine that maybe it's degrading kappa and then having empty heavy chains is leading to their degradation in some other way? So it's really this organism is degrading kappa, but as a result, the heavy chain is susceptible to other types of degradation.
That does make sense. And they do various experiments to understand what types of proteases may be doing this. Structural dependency. They do 3D structure, and I think that is the summary of this paper, that you can have a bacteria that is a commensal microbe in the gut that is producing a protease that degrades IgA and makes these mice susceptible to pathogenic microbes, chemical-induced injury, and C. albicans was their other pathogen. So super interesting. Do humans have bugs like this? I think is one of the big takeaways, because it could help us understand susceptibility across populations.
I think the one piece of data that you didn't really talk about was they tried whether these bacteria could degrade the antibodies from other species.
Oh, that's right. Thank
you.
And
it did degrade species that were close to mice. So rats and hamsters and guinea pigs. But as you got further away, it didn't. And it comes back to what you just said, which is what reminded me, is that if there are any human, wouldn't see them in mice. So you'd have to look more carefully in precise human analyses, because the human bacteria probably wouldn't degrade the mouse antibodies. And the mouse bacteria we know don't degrade the human antibodies. So it would be some sort of co-evolved group of bacteria with that particular species that would degrade the antibodies in that species.
But it certainly could, make us think more about where IgA deficiency or selective IgA deficiency is coming from. Although now I think Steph has made me realize in this discussion, the sort of serum versus fecal IgA issue that we would still have to think about.
And I don't know if it changes when people get antibiotic treatment, right? So they were able to reverse this when they treated with antibiotics and got rid of that particular species of bacteria. So we don't know whether people might be IgA deficient and not know it, get antibiotics, and then now be not deficient anymore, or what. So we don't really know if it's or reverse.
Yeah, it's a great point. And I think so what I liked about this is they do give you this schematic where their screening pipeline could be applied to any species. Mm-hmm. So they collected fecal samples from mice, but you could do this from humans and go through this process where you're trying to identify IgA degrading bacteria, and then you sequence them and you name them after yourself, and So that's my snippet.
I've been looking at figure 6D, right? They make recombinant human and mouse.
Yeah.
And I'm looking at the third column, human heavy kappa human, right? I mean, without bacteria or there's nothing in the lane. So I don't understand what's going on there. Both lanes are blank, right?
It probably is that if you try to combine human kappa, I know why, Vincent. Okay, so they made a human heavy chain with a human kappa light chain, and they're trying to detect that with anti-mouse IgG. Mm-hmm. So you can't pick up the human antibody with the anti-mouse IgG. But if you
look at an E.
In E, there you can see it because it's got the anti-human IgG. So the human heavy chain with a human kappa light chain is detectable with anti-human IgG.
And there you can see when they add the bacteria, then it's not cleaved. It's not cleaved very much. A little bit. Actually, no. C is the cleavage fragment, right? So it's not cleaved at all.
If they put the mouse kappa light chain with the human heavy chain, it now makes it susceptible to degradation with the bacteria. I think that's the key piece of information. So again, suggesting that it is something about, I don't know, that would say it's something about the mouse kappa light chain that's targeted by that bacteria.
Yeah.
Rather than what you were mentioning before was a possibility that the lambda was protecting.
Right. That's
so
a good point.
Seems like the kappa is targeting for some reason.
And perhaps in this case, it is that kind of free heavy chain is getting degraded else in other ways, because here the human heavy chain can get degraded.
Correct. So it's probably something else when the kappa is not bound anymore because that got degraded and falls off, then the heavy chain gets susceptible to degradation. That's why we need multiple great minds thinking together, because then we can piece it all together. Exactly. They probably said that in their paper if we read it more carefully. No. But I think it's hard.
The other thing to mention or to ask, why would a bacteria want to degrade IgA? And IgA has been shown to keep, I mentioned it a little bit in the beginning, but to bind the bacteria, either specifically or non-specifically. IgA has a lot of glycans, and you can have these glycan-glycan interactions, but they actually harness bacteria in niches in the gut so that they don't over-proliferate. They don't take over an area of the intestine, and then good bugs can't use those nutrients. And if there's an overproliferation, maybe you can have penetration and sepsis. The goal of the bacteria is just to make more copies. And so producing a protease that cleaves something that keeps you harnessed is beneficial for said bacteria. If I could guess why it wants to do that, that would be my hypothesis.
I agree, because I think it's just maintaining some level of homeostasis that works for both the host and the organism.
Right.
So if the organism makes the enzyme that degrades the antibody and the antibody keeps the bacteria in check, it's that balance of make some of the enzyme degrade the antibody that allows the bacteria to grow, then the bacteria grow, make more of the enzyme that degrades the antibody. And so they kind of like balance. And
so maybe the host really wants to make more IgA. And that's why when you treat it with antibiotics and you get rid of it, it's like, finally, I can produce some IgA. And if it doesn't come back in the system, it maybe can keep any introduction of this bacteria at low levels down.
Yeah.
And you could also imagine that IgA could influence the rest of the polymicrobial community. And so the IgA presence could influence those helper strains as well. And so by degrading IgA, you make a better place for your helper strains.
Or a competitor.
Right.
Mm-hmm. One or the other.
For nutrients, as we know this requires.
I'd like to see if wild mice have this.
That would be really interesting.
Lab mice are always, they're artificial in a way, right?
Right.
So it would be nice to see if wild mice do this. Maybe you have a different bacteria or something, but if wild mice did, then that would tell us that this is really important.
Well, I would assume that given if you look at the evolutionary association and how much this is sort of mouse and mouse adjacent organism specific, it probably has evolved to impact mice, and it probably evolved to impact wild mice. I think the bigger question is, are the wild mice healthy? Or are the wild mice, when they're not in an SPF environment, having difficulty?
Yeah.
Well, they're faster. I'll never forget one of my students years ago, she caught a mouse in her apartment. She said, "Do you want it?" I said, "Yeah, bring it in. I don't know what we're going to do with it." So she put it in a cage. And I put it was in the hood.
Oh, no.
lifted up the cage an inch, boom, it was gone. Holy cow, it just shot through in an instant. In a lab mouse, you could take the whole lid off and look at them and they,
I can't imagine.
Your Ayacook
Did
is
you tell
probably
the
thrilled with you.
Ayacook people?
No, I didn't tell him.
Oh,
It ran
no.
off into some crack in the building and it was probably living there
God,
still.
there's
no.
probably a whole family in there.
Whole family of mice now there, yes.
Yeah.
But it was just, there's just no way that I would have, or anyone would have.
Speedy Gonzalez.
Yeah.
Oh my God, it was relatively tiny as well. So
Also,
wild mice are incredibly fit compared to
Good
lab
word.
mice.
One more lasting comment. If you want to buy enzymes that were made to cleave IgA, there you can buy them. Various vendors sell them. That's actually what we use when we want to cleave IgA. We can buy them. So they've been isolated from bacteria that are not this one that we presented. So they exist elsewhere as well.
All right,
Cool.
thank you, Steph.
Yeah, thanks.
That was still fresh to me.
Good. Great.
I learned new things this time.
I love it.
That whole serum versus fecal thing
Yeah.
totally didn't come up last
Yeah, we
time.
did
And that's
hit on
like
that
really
last
interesting.
time. Yeah.
Well, if you have people who know what they're talking about, then you always come up with interesting things. Always.
Oh.
Yeah.
Like the other day, we did a TWIM, and Mark Martin says, "Oh, this is going to be a 10-minute snippet." I said, "You wait and see."
No such thing.
Oh,
So
you get people
it's
who are
hard,
interested. They ask questions, and it goes on.
Yeah.
So that was interesting. That was about. Did you know there's a China space station?
No.
I didn't know this. So they have their own space station up there.
I think all major countries do, right? Doesn't India and we do and Russia? I just
I
assumed.
thought it was
I think we
together.
share with Russia.
Well,
Yeah, I thought
ISS
we
is
shared.
made
Yeah.
from parts.
It's called the
of different
International
countries, right?
Space
Oh,
Station, right? To do it together.
Yeah.
That
I guess the
brings
more
up-
important question is why on TWIM were you talking about a
Because
Chinese space
they isolated
station?
a bacterium from the walls of the China space station.
Really?
Oh, wow.
It
Okay,
was living
that's
there,
cool.
right? So it was obviously brought up from
Yeah.
the ground, but it had been
Wow.
living there for some time. And so it was slightly different from what you find on the ground, right?
That's
Wow.
neat.
The question is, could it degrade IgA?
Yeah, right.
Well, so because I have a little bit of a advantage here in that I've heard Vincent tell this story on TWIV, I will say, Vincent, can you tell us how they did the swabs and how they did the controls?
Okay. So they swabbed the walls. First of all, they swabbed the walls of whatever is on the ground that brings astronauts up there, right? The rocket, the capsule. They swabbed that because they want to get a baseline for what's going up. And then they also swab the station. What they do is they take these swab, I guess they're cotton or something, in a plastic container, and they tear the plastic and they wipe the wall, right? And then they put it in something. But the control is they take the swab and wave it in the air,
Oh,
right?
I guess because it has to pass through air to get to the wall.
Yeah, but you see what's in the air if that's what's on the wall. But then they had a third control, which was they left the swab in the plastic and waved it.
Oh, really? Oh, as if
What?
the wave.
Just the
Okay,
act of
that's
waving
silly.
is somehow
That's
making
silly.
the
The third
bacteria.
control, but I guess that could just be your cotton control, right? Like whatever bug could be on.
Yeah,
the cotton.
that would be the
They
But it's
didn't
funny
need to
that they
wave
didn't
it.
have to wave it, right?
No.
Maybe they were having some fun.
Yeah. It's funny.
But it's interesting to think, is there different bacteria floating around in the air in space than there is here because there's no gravity?
Yeah.
It's the
I mean,
bacteria
I don't
floating
know.
around.
What could live up there? It's just
I
no
don't
air,
know.
right? Well, I guess they don't
There's
need
air.
But there's lots of ionizing radiation and all that,
Yeah,
right?
the-
That-
Well,
the
it's the same
radiation-
that the humans are getting
I guess
exposed
it
to
could
in the
get
space station.
resistant to it. Yeah.
You know.
Yeah, the radiation's a bigger problem. I mean, we have anaerobes here. So
Maybe they should have swabbed the outside of the station.
as Jeff Goldblum said in Jurassic Park, life finds a way.
Life finds a way.
Life finds a way. Yes. Yep. Okay, so I have a paper from Cell. It's about Zika virus. The title is Differential Contributions of Fetal Mononuclear Phagocytes to Zika virus Neuroinvasion versus Neuroprotection During Congenital Infection. Two first authors, Mohammed Abdelbasseh and Wilfried Saron, and the senior author is Ashley St. John, who was on TWIV I had visited Singapore some years, 2019.
That's cool.
And she was on a TWIV that I did at the National University of Singapore, which is where many of these authors are. And I picked it because we used to work on Zika virus. And in fact, we published a paper which should be cited here. And I'm very sad that they didn't cite it but it doesn't totally agree with their findings. So either they don't believe us or they didn't want to.
Just could have, but I like to think of the best in everyone. It just could be an oversight.
Could be an oversight also, but it's very hurtful for us.
I know.
But not that hurtful because I don't do any work on that anymore.
But still, you'd like to see it.
I
thought it was right.
I feel like the last time we talked about this, we came up with some like hypothesis of how both could be true, but I don't remember what that hypothesis was. So
we'll get there.
So the point, well, first of all, Zika virus, as you know, is a plus-stranded RNA virus, single-stranded, enveloped, which is transmitted by mosquitoes. And the issue with Zika is that around 2015, it suddenly started causing outbreaks of microcephaly in Brazil, which had never been seen before. And so it obviously can transmit from mother to fetus and causes this syndrome more than microcephaly. It's called congenital Zika syndrome. It's a multi-organ disease. And that brought a lot of attention to Zika because Zika had never been associated with congenital Zika syndrome before. So the virus was first isolated in Africa, and it circulated in Africa and parts of Asia without any indication that there was microcephaly. And then suddenly in 2015, it made its way to Brazil from Asia through the Pacific, and suddenly outbreaks of this CZS.
And there has sort of always been this question of, did the virus change and suddenly acquire this ability?
Yeah.
Or is this something that has always been a property of this virus, but maybe wasn't observed in, say, smaller numbers of cases that were being seen?
Yeah,
it was only until this larger outbreak happened that this rare event was really reliably cataloged.
Yeah. So our paper basically said, if you look at isolates in Africa from 1947 when it was first found to 2015 in Brazil, they all can infect the CNS of mice and cause disruption of neurogenesis. So it's different from what they find here, as you will see. But I'll talk about ours later. So the point here is that, all right, so they want to know what's going on with the Asian, African versus Brazilian isolates. And a little bit of background on, they use a mouse model and in early gestation, and that's when the risk of congenital Zika is highest. There are limited numbers of fetal immune cells present, and those that are there are primarily of myeloid lineages, and within the brain itself, they're mainly microglia, starting at day 9.5. Now, so the gestation of a mouse is 18 days, and 9.5 is when you first see microglia. And so these are derived from primitive macrophages that come from the yolk sac. Got to remember that, because that's going to come back to haunt us. And these primitive macrophages also give rise to tissue-resident macrophages in different places, and a portion of these go to the brain to establish local populations. And so because these microglia originate in the yolk sac, the microglia are present before the blood-brain barrier forms, which is not till embryonic day 11.5 to 15.5. So those cells are present at day 9.5. So they can sneak in before things shut down in the brain.
Yeah, and that timing becomes really important of, are we looking at infections that happen before that or after that?
So remember, the risk of this congenital syndrome is greatest in the first trimester or so. So that coincides with these cells being able to get in. Now, what do these microglia do? They do a lot of things during development. Of course, they are associated with injury repair in the brain and other tissues. In the brain, they do synapse pruning and picking up any debris from dead cells. And so here they want to know what the role of these fetal immune cells are in defending against Zika virus infection. So first, they take a collection of Zika viruses. They have Asian lineages, and those are the ones that ended up in Brazil and caused microcephaly, and then African lineages. And they say these strains were chosen since Asian lineages have been associated with CZS in humans, but African strains have not been. And again, it gets back to the issue of they have not, but maybe nobody looked, right? So many Brazilian physicians told me on TWIVs, there's such a background of bad health in many parts of the world, and there's a lot of microcephaly. You would have no idea if a virus was causing it unless you did diagnostics, and they don't. They don't do that. Anyway, so they infect these mice. What they do is they have immune-competent mice, and they infect the mothers, the dams, and then the virus gets transmitted to the fetuses, and then you can look at what's going on in the fetus, right? And so the first one of the things they do is they just measure the mass and head circumference of the fetus. So they see a reduction in mass of fetuses when the mothers were infected with Asian lineage strains, but not African lineage strains, less reduction in body mass. And they also find the head circumference. Now remember, microcephaly is small head, so the head circumference is a way to measure that. It was reduced when they used the Asian and African strains, both, but the Asians did it more. It reduced it more. So, this phenotype is not absent from what we see so far in the African lineage strains. It just seems to be less. And that's actually what they work on in this paper. They look at severe versus moderate disease, which is tricky, right? Always. It's better to have 0 and 1 rather than 1 and 0.5 or something like that. They measure the cortical thickness, which is also another reflection of microcephaly. Both strains induce a significant reduction in cortical thickness, but again, it's more severe for the Asian lineage strains. They also measure the placental mass to see if there's been any placental insufficiency during this infection, and they all reduce placental mass, all the viruses from Africa or Asia. So apparently, only once it gets into the fetus do you see some difference. So basically, to summarize this, both Asian and African lineage strains induce microcephaly with different degrees of severity. So they're going to basically study mild versus severe disease caused by the same virus. All right, so let's go through some of the experiments. They just confirmed that all these isolates can vertically transmit to the fetus. So they infect them and then quantify infection burden. And they found, and this is the other criticism I had last time, they're doing PCR all the time, not infectious virus. So you have to take that with a grain of salt because different viruses can give you different amounts of PCR product, but the infectivity is the same. Okay, you have to be very careful.
Yeah, it's good. Just, always throw an infectious viral assay into
why not?
your work.
It's not so bad, right?
They're fun.
Zika, you can work at BSL too, so it's not like SARS-CoV-2.
From your experience, is there something about a Zika plaque assay, or are they difficult do they make plaques? No, they're fine.
First time Amy tried it, it worked.
Okay.
It's great. Yeah, beautiful. All right, so they find Zika virus in 60% of the fetuses, no differences in the rate of PCR positivity, African versus Asian lineages. But in the PCR positive, so that's just numbers, but in the PCR positive embryos, the infection burden, so I guess the amount of genomes, the number of genomes was higher in the Asian versus the African infected. But the no difference in the placental burden. So only once it gets into the fetus, apparently by PCR, you see more product in the Asian compared to Africa. So I would say that you have to be careful there because you got to do some kind of infectivity assay. But nevertheless, overall, it's a very nice study. So I'm just pointing out little things that I would do differently. All right. Then they did they measured viral protein and nucleic acid in sections of tissues to just confirm what they've already found. And they can find Zika replication products in the embryos exposed to all of the strains. And they find antigen at some points, but those embryos were PCR negative at that point. It was like day 18 and a half. They find antigen, but no viral RNA is present. So this is a word of caution to everyone. Just because there's antigen doesn't mean there's virus replication, okay? Many people find SARS-CoV-2 antigens, for weeks and months after, and they go, it's persistent infection. No, it doesn't have to be. But look for infectious virus, okay, if you think it's a persistent infection. The antigen can hang around. I even learned from you guys that it can hang around in follicular dendritic cells,
That's
right?
Yep.
So, and this is in the brain. So protein can stick around. Protein is sticky. Okay. So these in all these experiments, they find replication and transmission and higher burden with the Asian lineages. That's what they see. All right. Next, they want to look at the cell types that are infected. And they use a number of markers, with the CDs in front of the number. And they say two primary clusters of cells that have Zika antigen in them. So they look for Zika antigen plus some other marker. And one cluster looks like microglia. And another cluster looks like infected neuroprogenitor cells. So they don't have any hematopoietic markers, but they have this marker called nestin, Nestin, which is a marker for neuroprogenitor cells, although it's a marker for other things as well, so it's not absolute. And but they found that 2 African strains result in a higher percentage of microglia-like cells in the brain.
Infected microglial cells, right?
Infected, yeah.
Yeah. So it's suggesting that there's a preference somehow for the virus to replicate, assuming it's replication, in different cell types from the different lineages, and they think that might be responsible for the different phenotypes.
Yes, and also the Asian and African differ in the amounts of. They're looking at viral protein here that can be detected. They can find protein in more nestin cells and fewer CD45. I guess those are the microglia-like cells.
Yeah, those are immune-related cells.
In Asian.
They color coded this, which I thought was really nice, but then I kept getting switched back and forth. I think the green is the African one.
And they also see fewer nestin positive cells in the brain of fetuses infected with Asian lineage compared to African lineage. So in other words, the Asian lineage seems to be depleting neuronal precursors in the brain. So the bottom line here, Zika affects both macrophages, microglia, and neuronal lineage cells, and the proportions differ depending on African versus Asian strains. Next, they want to know if the reduction in these neuroprogenitor cells is due to apoptosis or just viral infection, which is something exactly we tried to do with our system as well. And they find a greater percentage of apoptotic neuroprogenitor cells with the Brazilian isolate compared with the African isolate. And so there are more living NPCs with the African isolates compared to the Brazilian. So somehow these are surviving, these NPCs.
So some things somehow the NPCs in those brains infected with the African lineage are surviving.
Right.
While the neuroprogenitor cells in the brains infected with Asian lineage strains are dying.
Yep. And they find more phagocytosis of these dying NPCs in African lineage infected embryos. More efficient clearing when you're infected with an African versus an Asian lineage. So they say these can be infection targets, but microglia are also clearing infected cells from the brain environment.
And so that is another sort of caveat to using protein or using antigen as your measure. The phagocytic cell is going to have protein from phagocytosing viral infected cells, but is not going to be infected itself.
Right.
So they think microglia are involved in clearing NPCs in the African lineage infection. So they want to do an experiment where they deplete microglia from the fetus and ask what happens. All right. So they have a way of depleting it with an antibody. And so they're basically depleting primitive macrophages, the ancestors. So depleting those completely protects mice from infection by either African or Asian lineage strains. And the burden, the amount of virus in the dams didn't differ if you took out the precursors in the fetus, but the fetus almost complete protection. So
they really care here about those different types of macrophages, the ones that came from the yolk sac and the ones that didn't.
Right.
Sort of the early ones or the later ones. So they are looking here at specifically using a depletion before the early ones went into the brain. This depletion is specific for the early kind, not necessarily those later kind.
Yeah, the yolk sac macrophages. They also confirmed that these macrophages were being infected by staining, and they find that infection of these primitive macrophages precedes their migration into the fetus. So they get infected before they go in.
That's really interesting, right? Because I wondered, because I don't know that much about the other infections that cross across the placenta and infect the fetus. Do they all really act very early, or are some of them later? So, because this is presenting a model where the infection goes from the mom to the fetus, but basically to the yolk sac, and then those cells that are infected migrate to the brain, and then that causes the problem. So this really has to be an infection that occurs very early on during gestation in order for this microcephaly to happen.
So in other words, if the mom got infected in the third trimester,
presumably it would be okay. Yeah. But some of the other placental transfer ones, don't they cause abortion or things? Are they later in infection?
Yeah, I was going to say, so CMV, when you have later stage infections, the sequelae can be more serious. I think syphilis, I actually think syphilis at any time of pregnancy, but depending on the infection, I think it can.
Yeah, I think we'd have to first limit to placental derived things that lead to neurologic issues,
right?
Because it's really about the sneaking in past the blood-brain barrier.
Fair.
I don't know enough about the torch pathogens in general, and I definitely don't know enough about just the ones that cause neurologic issues.
No, I don't. So but this is a really neat Trojan horse mechanism.
Yeah, of course.
Yeah, it is.
Yeah, I mean the microglia are going in there to do their job, and then they bring virus in with them.
But I would assume that this is all kind of like by accident. I'm not that viruses do things on purpose, but I can't see a logical reason how that would lead to, the virus's goal is to make more copies and to infect new people so they can make more copies. And this doesn't seem to be a way to do that.
Well, I mean, it's just I would say it's another cell type to replicate in, and it just gets
stuck.
Yeah, I agree. Any CNS infection is an accident. It's a dead end because it can't go to someone else.
Well, okay.
I mean, it does
what about toxoplasma infecting mice? That makes them more docile?
Let's talk about people. Just people.
No, okay, fair.
Because we don't eat each other.
No,
I do not think pathogens at you.
Yeah. So, but people, any virus that goes, like poliovirus, goes in the CNS in 1% of infections, and it stays there. It's the end because it can't transmit. The way it transmits is from the gut, right? And Zika virus transmits via mosquitoes. So getting in the brain, I mean, it finds these cells, it can infect them. It gets into the fetus, and it reproduces in the fetus. But anything that goes in the brain is not going to contribute. And by the time the fetus is born, there's no more virus anyway, so it's not going to contribute to transmission. It's not a persistent infection.
Presumably, whatever allows it to get into these cells maybe allows it to get into some other cell.
Yep.
Yes. Then it's in the mother establishes a viremia, right? And that, I think that can be picked up by mosquitoes.
That should be the way it,
it depends on the level. Anyway, they do another way of depleting these primitive macrophage precursors and find the same results. Basically, these primitive macrophages probably promote dissemination of Zika via the natural route of it coming into the brain. They also want to put virus directly. They want to deplete these primitive macrophages and then put virus directly in the brain and see what happens. So when they do that, this is in contrast to the vertical transmission route, which we're saying, microglia, brain. Both of the strains result in replication in the brain when you directly put it in there.
Be very technically skilled to do fetal intracranial.
Fetal intracranial injection, yes. It's very tough.
But what that tells us, though, is that both of these have the capacity to do this, right?
Yes.
It's just that one somehow is able to hijack a ride into these early.
Yes. Well, it also underscores the importance of using an animal, right?
Yeah.
So we used, what we did is to take embryonic brains and make slices of them and put them in culture. So at different days after gestation, and infect them with Zika virus. And the way the brain develops is very interesting. So early on, there's a region of the brain full of stem cells that differentiate into neurons and other cells. And there are tracts of microtubules that emanate from the stem cell region to the periphery of the cortex. And the neurons migrate along those. And they make there 7 layers of neurons in the mature cortex, and those are formed by this migration on microtubules. And in microcephaly, those 7 layers are reduced in number. It makes the brain smaller. So what we found was that any strain from 47 to the Brazilian isolates could all disrupt these microtubules. If you put virus on the slices, it would disrupt them, and then you would not get neuronal migration. So we actually counted neurons that got to the periphery, and it's greatly reduced when you infect them. But both kinds of strains would do it. But that's consistent with what they find here.
Yeah.
You put the virus directly in the brain.
Yep.
So in fact, we don't contradict, and they should have cited us.
Well, and that's why I think it was just an accidental mishap.
You're such a kind person.
That's my nature. But I do think that totally agrees with, it is consistent with your work. And how cool is that?
Yeah.
Yeah.
So you need to have those microglia present to really see the difference between the Asian and African isolates. Yeah. Actually, I didn't think of this last time. I only thought of it this time going over the paper again.
Well, see, so it was destiny that it somehow magically didn't record.
Hopefully it will record and we don't have to do it a third time.
I know, really.
Can you imagine?
We've had so many aha moments.
Yeah. But then, okay, let's think about what are geographical or genetic conditions that could impact placental development, that could change the way a virus is able to access the brain, because we're understanding that its ability to access these cells, that is the differential factor. And I don't know enough about placental development specifically, but the genetics of individuals in Brazil and Africa are quite different. So that would be interesting to understand.
And nutritional status.
Yeah. Mm-hmm.
So another thing they find is that going back to mice, the original model of vertical transmission. So this is a tough one. The infected Nestin positive cells, the neural precursors, infected with the Brazilian isolate are more apoptotic than infection with the African isolates. In fact, signs of neuroinflammation were higher. In contrast, microglia are the dominant cell containing Zika antigen after infection with African isolates. So it's different. And these cells are highly neuroprotective in the context of both strains. So basically, the microglia can also be neuroprotective.
But those are not the ones that came from the yolk sac. Those are the later arrivers. The early arrivers can carry in the virus, and this seems to be particularly detrimental. The late arrivers can also carry it in and maybe are not so detrimental.
So they use a different model to deplete these early microglia in the fetal brain. They deplete them, and they show increased Zika infection in microglia depleted fetuses compared with their litter mate controls. So the microglia, that's when you inject virus right into the brain. So the microglia are doing the thing. Let's see. I don't want to go. There's a lot of experiments.
Yeah, there's a lot of data.
They also look at the influence of monocytes that are recruited into the brain. So later, they're recruited once you have infection, they're recruited in, and they want to determine the contribution of those to Zika infection. They basically find that the recruited monocytes contribute to non-protective neuroinflammation, in contrast to the protective contributions of the microglia to infection clearance.
Yeah, and so I think the idea was that, we're getting inflammation with the monocytes, and that's damaging the brain.
Right.
And maybe damaging some NPCs, while with the other cells, they're maybe clearing up the infected cells, but not leading to other bigger picture damage.
Right.
Well, the microglia, that's their job, right? They should normally be around. They should be relatively quiet and clear things out that are problematic. And that keeps the inflammation low. But if it gets bad and the monocytes come in, then they can drive the inflammatory issues that can then lead to additional problems, right?
All right. Next, the obligatory single-cell RNA sequencing. So they take these fetal brains and they dissociate them into single cells and do RNA-seq. And they say, what kind of genes expression is being changed here? And they can see a lot of pro-apoptotic genes going up. And they say it's more with Brazilian than with African. And so that's one thing. And then the other is that, wait a minute, there's some, all right, so pro-inflammatory genes with either one, although the African is always higher, the Brazilian is always higher than the African one in multiple lineages of cells. And they they have, they do this pathway analysis of the, that's in organoids, sorry. Anyway, basically the single cell transcriptome shows increased inflammation and depending on the virus, but also proteins that are involved in cellular communication among various cell types. And they say this suggests functional protection from Zika infection by microglia involves transcriptional paths towards highly inflammatory states and more cell-cell communication. Then they say, this is all in mice so far. So they do a few experiments in human organoids, brain organoids, right? And they ask whether microglia can be protective. And they make two different kinds of human organoids. And first, without microglia in them, right? Because when you make these organoids from stem cells, there's no microglia. They have forebrain and cerebral organoids. And they infect these and quantify Zika genome copies. And again, the Brazilian strains, the Asian strains, cause increased infection compared with the African, and both the forebrain and the cerebral organoids. Then they add microglia to these organoids. And this shifts the transcriptional profile of these cells. And so you can do single cell seq with these microglia. You can infect and then say what kinds of genes are upregulated by asking. There are programs that look at your transcriptional results and say, these are part of this pathway or that pathway. And the ones that are enhanced are in pathways called morphology of the head and microcephaly with neurodevelopmental impairment. So these are these conditions have been quantified in terms of genes that go up or down, and so these organoids respond that way. Also, necrosis and apoptosis genes are involved as well. Anyway, but nevertheless, what does Zika do with and without microglia in the organoids? So both African and Asian viruses were reduced when you add microglia, and you get greater protection with the African strains versus the Asian strains. So this confirms that the microglia are protective. Okay, so that's the data. A lot of data here. Fetal microglia are protective in vivo. They promote viral clearance and limit inflammation. But the early microglia that come in before the blood-brain barrier forms carry Zika virus in with them. So one thing I didn't mention is that they wanted to see if they could do some therapeutic treatment here. And so there's one of the induced genes is nitric oxide synthase, right, which would have an antiviral effect, but also damages tissue. So they have an inhibitor of this nitric oxide synthase, which they add to their is this organoids? I don't remember. They give it to the dams along with virus, and it reduces apoptosis, which is what you would expect, and so they think this could be possibly used to block monocyte-induced neuroinflammation.
Yeah, so it's definitely part of that later stage of cells coming in to deal with the virus, those later monocytes causing problems. So sort of like the early cells that I know very little about coming from the yolk sac seem to bring the virus and allow damage early on.
Right.
And potentially later cells, later microglia may be able to clear and sort of be more protective. But if we get monocytes coming in, then we get a lot of inflammation.
So their last sentence is, trafficking of primitive macrophages is hijacked by Zika. This is an accident. It's not hijacked. To disseminate into the fetus, their descendants are protective. So the descendants of those cells are protective. And non-inflammatory apoptosis of NPCs and microglia-activated viral clearance are key for neuroprotection during congenital infection. So a lot of principles here, I think, in this paper. Okay, that's it.
Yeah, it's great. I think it's such a problem. And we, in regards to Zika-induced effects, and I just think about when is the next time we're going to have a Zika epidemic, because they come and go so quickly, assuming population immunity increases.
Yeah, that's what it is, right? It's population immunity, since we don't have any vaccines. So
you don't really have a human cohort of Zika-induced microcephaly to test a small molecule type of drug. So, I mean, what I would love to see, because the monoclonals have been advanced pretty far for Zika in terms of safety, but they've tested this in rhesus macaques. I think you have a epidemic of Zika, any sexually active woman should have access to a monoclonal, because they have to have it circulating before the mosquito bites you and transmits. Because first trimester is so challenging to prevent any type of infection or impact, because you don't often know for a long time, especially in low and middle income areas, you might not have access to pregnancy test kits. So I'd love to see monoclonals be used as that in that way. But
yeah, I mean, I think this also potentially makes a case for vaccines or something like that, just because it shows that the time period is so important of
for sure.
The infection during that small window of time when the yolk sac cells are going in and seeding the brain, and infection that happens during that time is particularly problematic. And so anything one can do to make sure that there is no infection at that time, either, as Steph says, by using monoclonals or sort of by vaccine, whatever you can do to sort of make sure that that one little window.
You're right.
And
I guess we talked last time, monoclonals would be great for giving a high dose of a potent effector molecule. However, you have to IV inject someone, which might not have the stuff to do that. So then a vaccine really isn't the public health intervention that you would have to.
We talked last TWIV of a dengue vaccine, which is made of E protein subunits. That's the glycoprotein on the surface of the particle. And they modified it so it would be dimers, which is the way it should be. And the previous vaccine candidates were all monomers. They didn't work very well. And so they say this could be applied to any flavy like dengue and Zika. And so that could be an option, a protein-based vaccine.
Now, that dengue vaccine, though, would still have issues with this dengue serotype.
No, it wouldn't, because you can control the amount of each of the 4 E proteins. So you get a balanced response. There's a problem with the attenuated. Okay. They all replicate on their own, and it's hard to control which ones do well and which ones don't.
And so then you get different amounts of antibody because you had different amounts of virus driving it.
So that's the thing. That's why this is an appealing approach, is because you would avoid antibody-dependent enhancement, right?
Yeah. Which Zika we don't worry about. Is that accurate? We don't see a similar...
Don't seem to see it, no. There's only one serotype, or 2. That does it, right?
I mean, I guess the question is, I think the data was less clear on Zika versus dengue enhancement.
Yeah, I mean, there's some concern that Zika might enhance dengue, right? But I don't think that ended up being the case.
Interesting.
Well, the thing is that this, the situation in the U.S. with vaccine research is very unclear at the moment, right? Mm-hmm. And not just research, but providing such vaccines to nations that can't afford them. That used to be our role, and it's not the case any longer. And it's really unfortunate because presumably other countries can step up, but
it's short-sighted to think that
it's totally short-sighted.
We shouldn't bother with helping people elsewhere. But things like mosquitoes, they don't care about borders, and climate change, which is actually happening, changes the distribution of those mosquitoes that are now transmitting the diseases, and they're going to move up into the U.S.
There's no reason you can't be kind to others.
Yeah, that too.
It's so much suffering, right? I mean, that's the part that just guts me. It's just like so much suffering of people. So yeah, we have to keep pushing, because decrease in funding does not mean a decrease in circulating viruses. So
that's right.
Yeah, he says, let's give infectious disease research a break, but will the infectious diseases give us a break?
No.
I don't think so.
Absolutely not.
We got to keep at it.
That's Immune 92. You can send questions and comments to immune@microbe.tv. You can help us out, support us financially, microbe.tv/contribute. Cindy Leifer's at Cornell University on Blue Sky. Let's see, what's the address? cindyleifer.bsky.social, right?
Something like that, yep.
Something like that. Thank you, Cindy.
Yeah, thank you.
Steph Langle's at Case Western Reserve University. Stephanie Langle on X and Blue. Thank you, Steph.
Thank you. I actually recently deleted that Twitter account.
You deleted X?
I did. I might open a lab account, but I think for now I'm on the blue.
Okay, I get people telling me all the time to get off of X.
Oh, you can do what you want.
But I don't actually do anything myself. I have a few people who post podcast episodes. We have 190,000 followers. I figure it can't hurt to have.
Yeah, I noticed that when I was sort of migrating over to Blue Sky, some of my accounts that I followed that were not US accounts still seem to be particularly active on X and not migrating. And so I still have like a lot of contacts or a lot of sources outside of the US that I'm reading on X, even if I'm not actually posting anything.
Right. Yeah. I think if it helps your science and your science communication efforts, then
that's what I think.
Go for it.
Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
And Vincent Racaniello. Microbe TV is on Blue Sky. It's a Microbe TV account. It's Microbe TV, one word without the dot, microbetv.bsky.social. So people always say, you should go on Blue Sky. I say, well, we are. Just not me. It's the whole podcast organization. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month. I don't want to push the stop button now.
No.
Can somebody else do it?
I'm not going to touch anything.
No, I'm not touching anything either.
Okay, so there's a button here that says stop. Okay, I'm going to click it.